Good evening, this is Ranjit Singh Malla. Today I am recording my first ever video about machine learning and AI essentials. So in these videos I will be covering different machine learning packages and how machine learning can be used in different aspects and in the real life examples. This is the time for AI and deep learning. I have master in AI and machine learning and also PG diploma in AI and machine learning. The degree is from LJME UK and the diploma is from IIIT Bangalore, India. So the first ever things which I am covering for my online videos is about making deep learning models for ImmApex. So first of all, what is deep learning? How we can define the deep learning? Deep learning is basically a class of machine learning methods that is based on artificial neuronal network with multiple hidden layers that automatically learn feature representation from raw data through successive non-linear transformations. There are many definitions available from the textbooks, and this is one of the simplified version I am presenting here. Deep learning frameworks, how we can define them? One of the core building blocks. What are the tools? So the tools like, for example, TensorFlow, PyTorch, Keras, and what are they used for? For example, TensorFlow is used for end-to-end deep learning plus production model. Example is CNN for image classifications. For example, we are uploading lots of images on Facebook, on Instagram, and for example, YouTube, thumbnail, video, and many other photos or images we are taking. For example, we are taking images from tissue histology. So like TensorFlow is one of the important tool to classify the images in medical science, and for example MRI, which are very complex to classify. Only the expert can infer what is the problem in the brain. And so these deep learning models are getting better day by day for the image classification. For example, PyTorch, which transform model for natural language processing. For example, text generations. We are writing on the research paper and we want to modify the text. That is one of the example how it works in the background. Keras, for example, simple feed for network for tabular data. Like for example, we have a lot of data, sequencing data, origin expression data in the tables. So that's how we can use it to infer the important information. Or for example, there are lots of data coming from the banking sector, or lots of data are generated in different fields. So that is where Keras based deep learning framework are implemented and used. JEX is large scale optimization for neuronal network. Model architecture and machine learning libraries. For example, Hugging Face Transformer, Detectron 2, OpenCV, and PyTorch Lightning. For example, BERT is used for sentiment analysis, like how people are visiting to the Amazon and what are the customer sentiments. So those kind of things. Object detection, like Detectron 2, like people are passing through a traffic light, or somebody is entering in the hospital, how many people are going. OpenCV is, for example, real-time face detection from webcam and tracking the people, like on the city, like there are lots of hundreds of webcams, and that is kind of it can be used. PyTorch is for training ResNet with minimal code, like you need not to be expert in that aspect. Training, machine learning and development tools, MLflow, Weights and Biases, ONSX, Docker and Kubelocks. For example, MLflow compare accuracy across 50 models and runs, like when we are comparing multiple models and want to infer which one is the better. Weights and biases, for example, plot loss accuracy during the training. Docker is deploy machine learning model as preset API, and Kubeflow is example of end-to-end automated training workflow. There are some cloud-based deep learning platforms. Google Colab, many of us might have used it I have heavily used for my training when I was doing deep learning courses. AWS SageMaker, yes, deeply used for fraud detection model at scale. Like in, for example, in banking sector, fraud detection is should be very efficient. It should be very timely in the milliseconds efficiency. Google Vertex AI is trend plus server LLM based chatbots. Azure ML, this is from Microsoft. Recommendation system deployment, like okay, customer needs to purchase something, what they should based on their visiting history. Paperspace gradient, it run PyTorch training remotely. So as a biologist, we are heavily relying on single cell RNA sequencing, gene expression, and VDJ immune reporter. That is where immunologists deal majorly with. So what are the deep learning layer that we can use in single cell RNA sequencing? One is PyTorch, another example is SCI, VI, autoencoders. So PyTorch predicts cell type from expression. Okay. So what are the cell types? It is a B cell or it is a T cell, and if it is B cell or if it is a T cell, what kind of subset it can be. SCVI is basically can integrate multiple scRNA batches, like you have sequenced from multiple platforms and different batches, because you cannot hold all of the patient on the same day. Third one is autoencoder, like it compresses gene expression into latent space. Like for example, you have 1 million cell and you are getting the expression of 5,000 genes or 2,500 genes. How many dimensionality there are? So you need autoencoders to compress the dimensity, right? And VDJ, like nowadays the sequencing platforms or the sequencing technology getting cheaper day by day, and they are getting efficient. So VDJ layer, where we can deep learning layer we can implement PyTorch, TensorFlow, transformation, CNN, RNN model, and embedding model. PyTorch can be used for classifying antigen specific TCR, TensorFlow predicting binding specificity, transformers like learned CDR representation, motif detections in the BCR. Like I am working on antibody discovery aspects. So for example, CNN, RNN models can be very important for people like me to basically extract the sequence features and predict okay, which motifs is important for binding to the antigens. And embedding model, for example, converts CDR3 sequence into the vector space. This is fantastic. So where does ImmApex sits in all of these aspects? You are generating sequencing data and you are taking them to the deep learning models to infer some important information. So ImmApex is sitting somewhere in between, like you are generating the data like raw VDJ raw sequence, then you do some analysis like Seryuth, ScanPy, SC reporter, those kind, and ImmApex coming after that. Like you have the data and then you do some feature engineering and encoding, and those feature engineered and encoded data then serve as an input for deep learning framework, for example PyTorch, TensorFlow, JAX. Then you do model classification, clustering, predictions. So what deep learning tool can ImmApex feeds on? It can, the tool basically PyTorch, TensorFlow, JAX, they are one of the best tool for developing these multiple models. So for example, PyTorch based feeding like classify antigen specific TCRs. TensorFlow based deep learning model can predict the BCR bindings to particular antigens, for example herpes antigens, and for example the some of the cancer antigens. JAX, large scale immune model development. Great. So what deep learning tools can ImmApex feed on? Continuing like CNN model, RNN, LSTM, PyTorch. Like tool we can use PyTorch or TensorFlow. It can detect motifs in CDR3 sequence, BCR lineage classification, or learn CDR3 embedding for containing Hugging Face transformation, SCGPT, gene former or custom transformer model. How ImmApex contributes to this? For example, in Hugging Face, embedding as an input token or features. For SCGPT can integrate VDJ embedding as auxiliary feature or custom transformer model like ImmApex encoding used as a sequence token or a positional feature. That is where ImmApex is sitting. So ImmApex output can be the input for deep learning, one hot encoder, tokenized sequence, right, for LSTM, and for transformation property encoder for machine learning parameters, and geometric encoder autoencoder, and building network GNN, mute space, gen augmentation, and probability matrix. So that is, I'm just have given some of the background about like where does ImmApex is sitting, and now I will go to actually the R Studio to learn like how we can actually implement our learning. So first of all, you have installed all of this library, right? I have run and tested this code. So you install all of these libraries in R Studio, and installing R Studio itself is very easy. So the second thing here is, because all of the human like VDJ genes which are known, that is where we are like I'm testing this ImmApex. So you should get the reference sequence from IMGT, that is the largest repository for VDJ database, basically the germline sequences. So download reference immune receptor sequence from IMGT and immune repertoire and deep learning analysis. It supports multiple species, like it have sequences for multiple species like human, rat, mice, and you can download the sequence in terms of amino acid or in nucleotide, right? So let's get the sequence. So for example, I'm downloading the TRB, or the region is variable amino acids. You can do here basically IG, and you will get the IG. Right. For example, for the immunoglobulin, you have basically the heavy chain and the light chain, and for example going to, and it will take some time, so you can test it with lambda the sequences, or you can also test with the kappa sequences. Yeah, it takes some time, but it's not that time taking process. You are basing downloading. So now coming back to original where I have, right? So you have downloaded basically the chains, right? TRB or TRA. I have tested all of them, right? Great. So now you have to format the genes. So format gene function basically standardize immune receptor gene names from different sequencing platform into consistent IMGT nomenclature. It supports multiple technology including 10x, adaptive, air formats for VCR, TCR, reported data, and it can format different receptor regions such as VDJ and C gene across many species. It adds standardized IMGT column, example VIMGT, and validation checks VIMGT.checks to confirm if gene names match the IMGT database. That is very much important, and this is basically the code. We are taking one of the example data, and now we need to infer the CDRs. So infer CDR to extract the immune receptor regions such as CDR1, CDR2 from BCR and TCR sequences. Use standardized gene annotation from format genes together with IMGT reference sequence from GATA MGT. It supports multiple sequence technology including 10x, adaptive, and air with amino acid and nucleotide sequences. If gene names do not match IMGT reference, the output becomes NA simplified gene names. Use the first matching allele and extraction. Let's run the code. We are evaluating basically the adaptive technology based reference sequence, and we are extracting CDR1, CDR2, and these are some of the example. Right, now we are going to generate and augment sequence sets. So generate sequence function basically creates synthetic amino acid or nucleotide sequence for testing, machine learning, and deep learning models. It supports adding custom preference, prefix, and suffix motifs for mimic biological receptor pattern. It allows control over the number and length of generated sequence for simulation experiments. It can generate realistic sequence noise useful for data augmentation and generative models. So let's run the code. And okay, so we are using suffix motifs YF or prefix motif CAS is basically the amino acid, YF is basically the amino acid sequence, and it's basically to generate nucleotide sequence instead of amino acid sequence. You can replace like A, G, C, T, and let's test that with A, C, T, and you are getting the motifs. Okay, this the length you can set, or the maximum length you can set. Then you can mutate the sequence, like for example for antibody engineering kind of work you need the mutation of the sequence. So you mutate the sequence, introduce control mutation into amino acids like such as LALA mutation, or nucleotide sequence for data augmentation or robustness testing. It supports tuning the mutation rate, mutation positions, and number of mutated output per sequence. It can apply mutation across the full sequence or only within selected region. It is very useful for simulating biological variations, expanding small data sets, and evaluating model generalization. Like you mutate the sequence, generate a lot of the sequence, and test your model. So your mutated sequence, number of sequence 1, position start 3, position and adds, and these are the chunks you are taking and mutating the sequence. Then now have to engineer for reporter, like calculate the frequency, compute residue frequency profile across aligned immune, what residues, how much they are, measure how often each amino acid or nucleotide is appearing across the sequence. It supports sequence alignment through padding, trimming with max dot lengths. It is useful for reporter feature engineering, motif discovery, and visualization within tools like ggplot2. Let's run the code. Position 4, what is the frequency sequence size 7, 10. Basically, what is the frequency of particular amino acids to come? For example, the amino acid name is alanine, arginine, like aspartic acid, cysteine, glutamine. What is the frequency at position 4, 5, 6, 7, 8? Right. Now you calculate the entropy, right? Measure sequence diversity at each aligned position in immune receptor. Quantify how variable or conserved residue are across sequences. It supports multiple diversity metrics including Shannon, Simpson, value, Hill number. And here, for example, I am just tackling Shannon. You can just change it to the other diversity. Hill 1, for example, we are taking Hill 2, let's do it back to the position and do it with Shannon. Now calculate property of physiochemical property of amino acids, right? Amino acids are basically the chemical. They are attached together and making the protein. So compute statistics such as mean, median, sum of selected amino acid property at scale. It supports like HLA factor and other biochemical description useful for feature engineering. In machine learning model, useful biochemical sequences. What is HLA factor? Is a numerical description that represents the biochemical and physiochemical properties of amino acids. They reduce amino acid characteristics into 5 main factors using statistical density reduction. These factors captures properties such as polarity, hydrophobicity, secondary structure tendency, molecular size, volume, codon diversity, electrostatic charge. In deep learning and machine learning, HLA factor converts amino acid sequence into continuous numerical features that model can learn from more effective than raw letters. Now calculate the mean HLA factor at any position, right? So for example, HLA factor 1, 2, 3 for scale, that is their respective position and what is their values. Now calculate gene use, like the variable genes, basically like there are 40 to 50 for the V region, 23D, 6J region, for like 5 for TCR, 68 variable, 2 diversity and 14J gene. So how these genes are used to make a particular VDJ sequence, either it is BCR or either it is TCR. If it is TRB, then it will be VDJ. If it is IGH, then it will be VDJ. But if it is IGK or IGL, then it will be VJ, and if it is TRA, then again it will be VJ. Right. You can use your own 10x file. Here I have uploaded my own data, right? I have some of the data sequenced and I uploaded it just for testing and training purpose. Yes, you can use your 10x data. You can use data generated from different platforms. That is where ImmApex supports multiple platforms. You can calculate the motif, like which part of this your TCR or BCR basically binding to the antigen of interest. That is where like most antibody discovering scientists need to focus. It can identify and count recurring sequence motifs in amino acids or nucleotide reporter. Supports detection of both continuous and gapped motifs, right? Motifs is in 3D structures, so it does not need to be continuous. It allows filtering motifs by length and minimum occurrence frequencies. It is useful for discovering conjured amino receptor pattern, antigen signature and sequence biomarker. So let's count for motifs. Like for example, motif STA, YF, LVY. How many times it is occurring? For example, YYF, it is occurring 57 times. Now calculate probability matrix. It generates a positional probability matrix showing residue probability at each sequence position. It captures the OLA sequence profile of immune receptor reporter or antigen specific clonotype, right? For example, if you are studying some autoimmune disease, what are these antigen specific clonotypes in that? Not every clonotype is antigen specific, but only some of them are antigen specific. For tissue resident B cell, there is a high frequency, but for in the cells which are taken from the blood, the frequency will basically drop. So we are calculating the frequencies and then we can generate a kind of logo of that frequency. Oh, this is basically how it looks: acidic, basic, hydrophobic, neutral in that particular region. A positional weight matrix is generated from PVM using log-like scoring against background residues. Right. So let's run the simple background frequency. Great. Now adjacency matrix, it calculates residues co-occurrence relationship between neighboring sequences, like what will be the next amino acid to this particular amino acids. It creates a matrix showing how often amino acid or a nucleotide appear next to each other. It helps analyze local sequence structure and transition pattern within reporter, useful for network analysis, motif discovery, and sequence relationship modeling. Let's run the code. Great. Now we are coming to building the network. So build a network basically creates a sequence similar in a network where each node represents a sequence and edge connect similar sequences. So basic use of workflow is basically Levenstein edits distance to measure sequence similarities. Let's run the code. And we have created basically the network. Great. And we have used the GG graph in the background. So let's create the standard TCR-BCR network. For TCR-BCR reporter analysis, normalized sequence distance are preferred because receptor sequence vary in the length. Normalization involves biological comparability between clonotype and reduce biases. Normalization basically improves biological compatibility between clonotype and reduce biases. It helps identify more occurrence immune receptor similarity networks and clonal relationship. Great. Let's run the code. We have generated the matrix. So finding clone type with V gene filtering. So we can filter basically the V gene and improves biological relevance because true clonotype usually share the same VG clonotype. Like for example, one particular expanded clonotype. So it heavy and light, and like there can be parent clone, one parent clonotype, and then there is a lineage. Like particular gene is used, then like heavy chain is constant or the light chain is changing across them. So something like the parent cell and then parental clone, and then there is a diversification during the like the development of the immune cell. Let's coming to the encoding sequence for model input. Now we are going to encode the sequence. So sequence encoder converts biological sequence into numerical embedding for machine learning and deep learning model. That is where we want. We have talked in the PowerPoint presentation. Okay, ImmApex is generating basically the input for deep learning model, CNN for LSTM, for transformer, for machine learning, for encoders, right? So let's run the code. Generate geometric embedding for each sequence. We are using the geometric mode. The method is blossom62, and we are keeping the theta pi 3, right? Then property encoder. Property encoder represents amino acid using continuous physiochemical features. Like previously we have talked about HLA factors. Great. And these are some model specific details, and if you want to read more about, you can go through these references. And then we generate a tokenized matrix. Then geometric encoder is basically converts an entire sequence into single fixed length embedding vector. Use substitution matrix like blossom62 to represent amino acid similarity numerically, right? And let's run the code. Property set we have done FSGI, and that is where we have tokenized sequences. So tokenization basically converts sequence character into integer token for machine and deep learning models. Each amino acid and nucleotide is mapped to a unique numeric ID. It supports adding start and stop token to mark sequence boundaries. And these are the model specific parameters: add start token, stop token, max length. Let's see our matrix head. Then sequencing decoder. Like in machine learning, there is encoder, then decoder. There are two parts, and the in between that basically the hidden layer is sitting. Sequence decoder is basically construct biological sequence from one hot or property encoded matrix. It converts numerical embedding back into amino acids and nucleotide sequence, useful for interpreting outputs from generative AI and deep learning model. It enables sequence recovery after encoding, simulation, or generating model. Let's run the code. We are using one hot decoder, right? Let's run the next code. And now we need to train the model, like classify sequence with random forest. Great. We are using the method of random forest, and basically SLA factors, all their physiochemical properties are taken into the care. The motif we have considered to CS or the CSG, right? And basically the random forest is being used to train the model, and then extract the important data from the model. Like a key advantage of random forest is its ability to easily extract features importance. We can create a plot to see which positional HLA factors were most influential in this model prediction. So these are the top 10 most important features. We have extracted and plotted them. Now we can use unsupervised clustering with PCA and geometric encoding. Unsupervised analysis can identify natural sequence cluster even without class label. Great. PCA reduce the dimensity into visualization component. Let's run the code. We are simulating and encoding mixed population of sequences like CA, QR. These are the motifs, and we are keeping the number of sequence to 100. The method which we are using is basically the Blossom 2. Now perform PCA and visualization of the cluster. We have run the PCA principal component analysis. 3 different groups, group B, C and A are basically the inputs, and these 3 principal, like how these 3 groups are separated from each other. Now identify sequence communities with network analysis. That is very important. So you can make small cluster and simulate data in errant structure. So now you are keeping the motif CSS, CRS, CSVA, and you can define like which motifs you want to build the sequence network. Let's build the network model, right? When you use Scanify, you also generate some kind of this networking graphs, and so you can correlate with like how these networks are generated based on the similarities. Now perform the community detection based on those similarities scores, and visualize and interpret the communities. Now convert into the graph, and we have to visualize all those community, right? These 3 communities, we have seen these 3 communities in the principal component analysis. Let's look at the PCA and looks at the graph. That is definitely looking much better than what is PCA. So this is my first ever video for the YouTube, and I have not done it second recording. This is my first ever shot, and I have recorded it in the single shot, and I'm uploading. And now I want to start interacting with people. I want to collaborate on deep learning and machine learning. I have MS in artificial intelligence and machine learning, and also the PG diploma, and I am happy to answer any questions you have. And I will uploading both the PowerPoint presentation and also the RStudio RMD file below my video, and you can have a look. If you have any questions, get back to me. Now I am stopping my recording. Thank you. Jai Hind.